consultative, and then everything else is broad spectrum. All the way down. This one's broad spectrum. So that just helps me remember. Yeah.
What's the relationship between method of action and
So used to treat, it just depends on what they've found experimentally to be effective. So yeah, don't remember that part. It changes a lot.
just method of action?
And actually, it depends. Just remember method of action. Yeah. A lot of that depends also on like practitioner specifics or generation of those drugs. They've come up with new techniques to get the pharmaceutical inside of gram-negative. So you know they're used to treat different things. But I just kind of threw some out there so you'd be like, maybe some are used to treat a STI or a UTI. Just kind of recognize, yeah.
And then what's there's a relationship between gram and method of action?
Well, not really. So gram, if you notice that there's only really 1 gram-negative, and they have a really hard time specifically getting the drugs inside of gram-negative. So even though those ones are broad spectrum, they have a hard time always treating gram-negative. So anytime somebody, you'll see it on the chart, is always gram-negative rod, then you go like, oh man, you're like, what do we do? So they're difficult to treat with all antibiotics. But yeah, the gram isn't really necessarily associated with the method of action. And because what they started doing with the broad spectrum is, yes, it has the molecule that will kill the bacteria itself, but it comes with other chemicals to help it get through the gram-negative membrane. So yeah, it's just you just have to memorize it.
So when it's attacking the 30S subunit, that's like denaturing the ribosomes?
Oh, for the ribosomes, yes. So the ribosomes, it does. So it will attack the ribosomes, but it has to get in first to get to the ribosomes. But yes.
So it does go through the cell wall, but it doesn't destroy the cell wall.
It doesn't destroy the cell wall.
Like the second one, it's just attacking the cell wall.
Yep. Yeah. And the way it does that is through the enzyme that forms the cell wall. Yeah.
So you really just have to know the class and what it does. There's not like these classes do this.
Yeah.
And metabolic path is that?
So that one is folate synthesis. So you'll see it written both ways. You'll see like metabolic pathway, but really it's this when they have to make folate and they need folate. So they make folate to, and if they can't make folate, then they'll die. So it's just that's their metabolic pathway. But you'll see it written both ways. So I just wanted to throw that out there.
And what is there like attacking the 50S versus the 30S?
Yeah, it's just the way they're done. Yeah. Bless you. And that's helpful to know because you don't want it to attack. We don't have 50s as subunits or 30s subunits, so it doesn't attack our ribosomes. Yeah. Okay, so just in the last 10 minutes, then we can go through these together. So these are aminoglycosides, cephalosporins, tetracyclines, penicillins, sulfonamides, the fluoroquinolones, macrolides, carbapenems, lincosamides, and glycopeptides. So those are the names of those. And then some of the examples, so we know the examples for cephalosporins, so you can put like Ceph or Ceph, something like that to help you remember. Tetracyclines would be cycline, cycle, something like that to help you remember. I should probably write it correctly because I would just have it for myself, but Okay, and then ampicillins would be any of the psyllins. Sulfonamides is the sulfas. And then ciprofloxacin, the floxacins. Floxacin. And then the azithromycin, the penums. So these are penums. And that's it. So those are the ones, the examples that we know. I'm sorry. And then really you only have to memorize 4 gram reactions because the rest are broad spectrum. Right? Everything else is broad spectrum, so you have to know those 4 that are those 4 exceptions. And then make some sort of use some sort of technique to help you remember the method of action. So 30, you can do, so 30, I'm gonna not have this correct. You could do cell wall. I would maybe put peptidoglycan for myself, just so I would remember that, but 30 sulfas are folate. Then replication of the fluoroquinolones, replication, and then macrolides are the 50, and then cell wall, 50, cell wall, macrolides, cell wall, 50, cell wall. Some sort of quick way of knowing. Here for our cell wall, these are the beta-lactams, so you can give yourself a little note of which one are the beta-lactams. This one's beta-lactam-like, but that's okay, we could just throw it in the beta-lactam ring. Replication, why did I put psyllins for 30? Am I missing one? Is that why? Oh yeah, it's because I put these too far apart, right? Yeah. Okay. So it goes cell wall, and then 30, and cell wall. Beta. Okay. So there's not very much to write down. As long as you can remember these, and I would write them down, and I would do this chart 10 times at least for myself. Yeah.
Do you want us to have like 1 or 2 of each of the examples memorized?
So this is all I would memorize. Ceph for the ceph triaxones, cyclines for the tetracyclines, psyllins for the penicillins, sulfa for the sulfonamides, floxacin for the fluoroquinolones, penums for the carbapenems. And so 1, 2, 3, 4, 5, 6.
So like if you ask us for an example and we wrote down penums, is that going to be sufficient?
No, I won't ask you. I'll say this. I'll say like ampicillin, and you in your mind you go, oh, psyllin, that triggers me. It's a penicillin. So you don't have to memorize how to spell those. You don't. Yeah. Does that make sense?
Yep. Thanks.
Okay. Yeah. Glycan. Oh. Yeah. So it's beta-lactam-like. The structure is enough like a beta-lactam, so sometimes it's thrown in there, sometimes it isn't. For the sake of our class, we'll just throw it in there. It's beta-lactam-ish.
So if you ask on an exam, if this is like considered beta-lactam,
yes, but I won't ask anything tricky like on the fringe like that. Yeah. Okay, so one thing I wanted to mention is as you're writing this, writing it to memorize it. You get your quiz on the back, you write it again really quickly on the back, so then you're like, okay, cool, now I have my little cheat sheet on the back of my quiz. Awesome. So that's a great way to memorize, but when you're a clinician and somebody says, oh, it's ciprofloxacin, and you're like, oh, that's a fluoroquinolone. I remember the flow. The floxacin is the fluoroquinolone. are you gonna remember it's a gram positive or gram negative without writing out the whole cheat sheet?
No.
No. So one thing to mention is it's a really good idea to know the chart, but if you are going into clinical medicine, take an extra hour and just go one by one and go like, okay, aminoglycosides, what are those? What do those, give me some examples. I'm gonna remember aminoglycosides. That's my only negative. That's my targeted negative antibiotic. Okay, what targets gram negatives? Aminoglycoside, negative, 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 negative. It works on the ribosomes. You know, actually when you get into clinical medicine, they're not gonna, I don't think they're gonna ask you, maybe nursing school, they might ask you what the target is on the bacterial cell. Or if you go into taking the MCATs or something like that, they might ask you. So it is a good idea to try and look at them individually and not just memorize a chart. And that's true from here on out. You know, I'm a chart person in my mind, and so I like charts and I can memorize charts easily, and if I put everything in a box, I could fill in all the blanks pretty easily. That's how my mind works. But that's not how it works when you're standing in front of your patient. So just make sure that you take a little bit of extra time to go like, okay, what is an aminoglycoside? I'm gonna know my antibiotics so well that I know each one individually and what they do. Because if you are going into nursing, or if you're going into PT, PA, MD, they will 100% want you to know this inside and out, backwards and forwards. So my girlfriend that's in nursing school was like, oh my gosh, this saved me like a month of work, because she already had it memorized. She already knew she could move way ahead of everybody else because she had it memorized already. So it will help you just to get the hang of them and already get to know them. Okay. So I'm going to take just a couple extra minutes before everybody else gets here. If you guys want to keep practicing, you can keep practicing. But I'm going to explain this one more time. So this is why it's hard for people who don't get here early. It's not anybody's fault, because it's difficult sometimes to get here early and you're doing multiple things. But you know how this was kind of difficult to explain yesterday, and I tried to simplify, but I was kind of like how do I make it so that you'll understand it? Well, they only got to hear it once. But that's the nature of an hour teaching you microbiology in an hour and 20 minutes. It's very difficult. So I'm gonna go through this again, and if you feel confident with this, great. Maybe share it with somebody else in the class who's struggling. So let's go through it again. This is just the beta-lactam example. Remember, they're not all beta-lactams. Oh, let me pull that chart up. This one. So here are the, so for DNA replication, that was the topoisomerase. So that's how fluoroquinolones work, is they attack the topoisomerase. So during DNA replication or transcription, that it can't unwind and read. So that's what targets DNA replication. Folate synthesis, so those ones work differently. Protein synthesis, those ones work differently. So this is just the beta-lactam example. So how we found those was in 1928 was Alexander Fleming. He found the lawn of bacteria and found a plaque, which is like a clearing, which we're gonna do that in the lab ourselves. But what it was originally the Penicillium fungi created this molecule that helped it to defend itself against bacteria. So a happy bacteria will have this bacterial transpeptidase enzyme that lays down new peptidoglycan. And then it can divide, it can go through binary fission. A happy bacteria is a bacteria that has a bacterial transpeptidase because it can lay down peptidoglycan. But this beta-lactam ring molecule will be all around, because this is spitting out these beta-lactam molecules, and because it's spitting out all those beta-lactam molecules, now it's going to be plugging all of the bacterial transpeptidase enzymes, and now it can't lay down peptidoglycan and the bacteria will die. It can't reproduce. It can't go through binary fission all the peptidoglycan will break down, and that's how beta-lactams kill. That's still, if somebody takes penicillin, that's how it works. Okay, so that's our beta-lactam example. Does anybody have any questions about that? Yeah.
What is that enzyme called?
This one is bacterial transpeptidase.
And that makes peptidoglycan.
That makes peptidoglycan. It forms and pulls all the pieces together.
So it means that does it always have to be creating that layer?
No, so that was a good question. I just kind of gave this as a little cartoon to help you see it, but really what happens is it can't replicate.
Okay.
So it can't lay down new peptidoglycan in binary fission.
Okay.
Yeah.
But that particular bacteria wouldn't die.
Yes.
From this.
It just
So
won't
wouldn't
be able to replicate.
Okay.
Yes, yes, great question. Yeah.
So where you wrote the penicillin fungi,
Uh-huh.
is there anything special about the way you're drawing here, or is that just how you represent the fungi?
This is just how it's just a cartoon.
Okay.
Yeah, yeah, so we'll learn about that. It has like sporangiospores, and we'll learn about the structure and all that kind of stuff. Now it just looks like a plant.
Okay.
Yeah.
Similarly on the ring molecule, are those lines significant? Do we learn about those?
Oh, so the beta-lactam rings, so what those are is, I don't know, do you guys learn that in Gen Chem, or do you learn that in organic? Oh, that's why. Oh, now I'm confused everybody. Okay, so I've been doing that for 5 semesters. I'm glad you brought that up, because I thought just because of the chemistry, everybody would know what that meant. But it's just directionality of a chemical, and I don't know how to draw the whole beta-lactam ring structure, and there's a bunch of them. So let me show you the different ones. So here. So there's something attached over here, and there's something attached over here. So it's just different, what are those called? Dr. Duffy can help me.
I gotta see on
Trans and cis.
Yeah, I forgot.
Yeah, like cis and
I
trans. Me too. I'm like, bring back into my, when I was 19 or something.
It's like a shorthand for the molecule structure.
Yeah, for one that goes one way and one that goes the other way.
That's
because they,
cis, isn't it?
Cis and isn't that trans? Cis would be the same.
I thought
Transphase is like exiting towards the membrane.
Trans is the
Transphase.
opposite. That's trans.
trans is the opposite.
That's
I
trans.
confused.
Okay, yeah. So they're just attachments that I don't know how to draw. Okay. Okay, yeah, here are the rings. Here's the different cephalosporins, carbapenems. Here's the ring that the bacterial transpeptidase, that it will
Is it always
block its active site.
is
of
it
the
always a
enzyme.
4-carbon ring?
It's always a 4-carbon ring. That's what the beta-lactam
I
structure is.
thought those weren't very stable.
Yeah, they're not. They're not. It's a rare. We can't really find them in other places. It's a natural antibiotic that the penicillium made. Yeah, they're not. They're hard to synthesize, and yeah, they're not stable. Yeah, good questions. Okay, awesome. Okay, so let's move on. So we talked about antibiotics. Practice your antibiotics over and over and over. Make sure that you're comfortable with our antibiotic terms, and we're going to talk about antibiotic resistance. So remember when we have prevention, identification, and treatment. Prevention, we kind of have a lot of tools, right? We have a lot of chemicals, we have a lot of mechanical methods of killing bacteria, of preventing infection. We have vaccines, which the vaccines aren't all the way amazing either, but we do have that as a tool. For treatment, if someone is sick with a bacterial infection, we have one tool, and that's antibiotics. So this tool has been, I think I showed you guys that picture, but I can show it to you again, of the clinic that I worked at in Africa, where, we had wall to wall to corner full of antibiotics, of expired antibiotics, of leftovers of 2 pills in a bottle, of all kinds of just crazy things. Some of them were not in English, and some of them were, just like all different, prescriptions of like random. I don't even know how we ended up with all of those. They were just donated. So what they're doing is when they're farming those out, which is exactly what we do here, by the way. We farm out antibiotics like you would not believe. So when you're farming out antibiotics, what's happening? Why is this tool not working anymore? What's going on with our antibiotics? Why aren't they working? So I always give this example of this person who sprays for bugs. So this person who sprays for bugs, for example, this is going to help you to understand what antibiotics resistance is and how it works. So what happens is somebody sprays for bugs and it will kill all the bugs, except there was one bug somewhere that had one genetic mutation, that had a point mutation, that created a protein that was a little bit stronger than the other bugs. So now this one bug has a little bit of a stronger exoskeleton accidentally because it had a genetic mutation. So this one bug survived. Now that this one bug survived the spray, now it has all the food and can reproduce. And now it's taken over and all of its genetic offspring all have the stronger exoskeleton. So that's a little bit of how like evolution works, but that's exactly what's going on with antibiotic resistance. So with antibiotic resistance, when somebody, I'll go through the different ways that antibiotic resistance can occur, but let's just use the same analogy. So let's say that antibiotics killed 99.999% of the infection. Well, whatever is left is now it's got all the food and it's living the dream and it's comfortable. And now whatever helped that one bacterium survive, now it can go off and reproduce, and now all of its offspring are stronger. All of its offspring can survive that. So that's how it works kind of in a nutshell. But let me give you some more specific examples. Oh, I love this video. I'm gonna have you guys watch it.
Except that it's 2 feet by 4 feet. And the way we set it up.
So he said this is a petri dish. So this is an agar plate. This is just like our agar, exactly the same in our lab, except for it's this big giant agar plate.
9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pours thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
So that's kind of terrifying, right? It's if you were to put that antibiotic or that bacterial culture